All right, so before I start here with the PowerPoint, yes, I did some modifications with the PowerPoint. I took out some slides. I added some clarification of some explanations, as usual. So I did put up the new PDF for you on the modules. Okay? And this week you are going to have your first quiz, so get ready for it. Now, the quizzes are not intended to be very painful. Okay, they're intended just to be educative, instructive, and mainly as a type of practice for exams, right? So I do allow you to have two, goes at the quizzes, right? So I think I'll give you plenty of time, okay? And you can try twice, so everybody can get maximum grade in every quiz. So the only thing you have to do is make an effort and take the quiz, okay? And make sure you take all of them because it's a good percentage of your final grade. Now, next class on Thursday, I will not be able to be here, okay? So I will hold you responsible for the class. I will have to use the recording for the last class that I did last term, okay? And the reason why I will not be able to be here is that, unfortunately, my health has not been the greatest lately. And this weekend was dreadful. And yesterday I spent the whole day at the ER. Yay, pumped up with morphine. If I look a little bit too happy today, it's because my brain is under narcotics. Legal ones. I was trying to avoid it as much as possible. But sometimes it's a kind of impossible. And why am I mentioning this here? Well, mainly because this whole havoc that my system is putting me up to is due to inflammation. Yay, and this is very relevant to the topic of innate immunity. So you gotta know about inflammation. And if I got knocked out, it's because inflammation is a powerful thing, okay? But we'll get there whenever we get there, and mechanisms of inflammation, I'll let you know. But anyway, on Thursday, I have to go to the only appointment that was available with my specialist, who is actually going to travel out of the country in the next few days and not coming back in a few weeks anyway. So I have to see him, okay? All right, so now, I did open the iClicker, so you do have some questions in iClicker, so get ready for those. And I do also have some questions in this PowerPoint that are for your practice. Okay, so those they look like iClicker questions, but they're not. They're just going to be for your for giving you extra practice questions. All right, so here, coming into our continuation of the overview of the immune system, because I know it's a lot of information and it's a lot of things going on, like at the same time. Okay, so it sometimes it's a bit overwhelming just to look at this big picture. So let's break it down into components and look at them, of course, like we have been doing individually. So we were talking before about those pattern recognition receptors. So when you see PRR, you should think innate immune cell. Okay, that doesn't mean that the innate immune cells are the only ones that have PRRs. Okay, you have cells in your adaptive immune system, like your B cells, that they also have PRRs. Okay, but those B cells have other receptors that are more important for the functions of those B cells. Right? So when you see PRR, you should automatically think innate immune cell function. Okay? Now, those receptors on the innate cells, remember, they are going to detect patterns, right? Now, they're going to see patterns, right? And the way they see patterns is by feeling, right? So they don't have eyes. They have to go around like that, and they go by feeling. Anybody watch Crocodile Dundee? Maybe that's not a good analogy, but anyway. So go by feeling, okay? So go feel, and you say, oh, I feel pattern here. Now, notice I use both hands. There is a reason for that, right? Why? Because if the guy goes with one hand and feels a pattern, well, it's not a pattern, it's just a molecule. Okay, fine, feel a molecule. And if by feeling only one molecule, this immune cell gets activated, that's a problem. Okay? So when these cells go around feeling these patterns, they go with more than one receptor. They have to use more than one PRR. Okay? So they go around, and they see a pattern here. This one detects a pattern. This one is like, oh, I didn't detect anything. It's like, so this one, only one signal coming from only one pattern recognition receptor is not enough to activate your innate cell. It needs at least 2 of those receptors working at the same time in order for the cell to be activated. The reason is that the patterns that these things are feeling, they are not unique to pathogens, okay, or to damaged cells. Those patterns can also be present in your own cells. Those patterns are usually made up of some sugar residues, okay, some saccharides. And saccharides are present in your own cells too. The difference is that when you look at cells that are microorganisms like bacteria, okay, those patterns show up a lot. So you have high density of patterns, okay? Now, when you have this high density, then the receptors, you're going to have at least 2 of them. Okay, so this thing is not very well explanatory. So let me draw here another pattern. Okay, so here, at least 2 patterns have to be activated for you to get some response out of the cell, okay? So this is a key feature that differentiates the activation due to some kind of microorganism that is an invader versus the lack of activation based on your own cells, because again, your cells are going to have this type of patterns, okay? And this type of distinction on how these things get activated, this type of explanation, a lot of times kind of fly under the radar because it's not very obvious when you read through these immunology books, okay? And usually what I see happening is like what happened to me, for example, guilty, okay? What happens, I read the book, I memorized it, and then comes to a test, I answer the questions properly, I pass the class, and then when I stop to think about it, it's like, why? And then I'm thinking, oh, I don't have time for that now, I'll worry about that when I have to. That's usually the survival mode that we usually get into when you're learning something just to survive and go to the next class or get a degree or something, right? But it's really, really important, like in the upper level class like this, it's really important that you start questioning why things are happening, how they are happening, what is the mechanism that is triggering a certain response or the lack of response. And when you're talking about responses of the immune system, everything is important. Okay, so this distinction of concentration or high density of patterns is really a key feature to understand why you can get activation of certain immune cells or not activation of certain cells. Okay, so these patterns, how is it the receptors? So how is it that they are working? So mainly you have 2 types of functions and 2 types of receptors that can work in detecting and doing something about whatever they detected, okay? So one type of receptor falls into this category of phagocytic receptors. So those are your phagocytic PRRs. What do? They bind to PAMs. PAMs are for those pathogens, right? They bind to PAMs, they eat the PAM, the thing that carries the PAMs, okay? They eat the thing and digest it, and they're happy, they got their food, okay? Now, these types of receptors, their main function is to cause phagocytosis to take place. So here is your phagocytic PRR. It bound more than one of these patterns, recognized them, and now it engulfed this bacterium, and it killed the bacterium somehow. There are several ways of killing the bacterium once it gets engulfed. Okay, so this is the job of phagocytic PRRs. And then you have this other type of PRRs. There are signaling PRRs. Okay, the signaling PRRs, they are going to activate some type of response in the cell, but it's not going to activate phagocytosis. So this signaling PRR is going to activate some form of intracellular signaling that is going to cause this cell to start producing some kind of proteins or toxins to be released in the environment. Okay, so phagocytic causes the thing to undergo phagocytosis, and then signaling will cause something to trigger exocytosis of some type of molecule. Okay, now what are the cells that have PRRs? Of course, you have your macrophages and neutrophils, so those are your main phagocytic cells. Okay, then you have your eosinophils, basophils, mast cells. We're going to see more on the characteristic personality of these cells. Okay, all right, so just a little side note here that once you have your bacterium engulfed and phagocytosed, then one option is for this phagosome, that is the initial vesicle. So this phagosome to get together with another vesicle that is called the lysosome. And lysosome has enzymes that are going to chop up the bacterium. Okay, so when phagosome gets together with lysosome, this vesicle here becomes the phagolysosome. Okay, and then we're going to see later next lecture on mechanisms of how to destroy the pathogen once it is inside of these phagolysosomes. Okay. All right, so how do you kill the pathogen? Right. So we know that your immune system is always watching. You know, that reminds me of a scene from Monsters, Inc. Everybody watched Monsters, Inc.? Yeah. You know, the old lady that was, I don't know if it was one or two, I don't even know how many of Monsters, Inc. were there. But she was, I think, like a secretary or something. And then it was Mike that was coming out, and she goes, "I'm always watching." You know, that woman, the monster, you know, that, yes, like that's the immune system. It's always watching, always. Okay, so how is it watching? Well, it is surveilling your circulation. It is checking out anything that comes into your system that's not supposed to be there. So circulation means your blood circulation, and your blood is going to circulate through your arteries and veins, right? And it's also watching your lymphatic circulation. And that has to do with the lymph that comes out of your arteries and veins junctions, right? And the lymph gets basically sucked up from the site through these lymphatic vessels, which you're going to see more of it, in a little while. Okay, so the surveillance is always around and is also in the tissues. Okay, you have some cells that like to sit in the tissues and watch what's going on and get ready for some fight if they have to. Okay, so basically there is not only one strategy to kill the pathogen or invader. You have to really look at how is it that your pathogen has to be killed. Sometimes you can eat it, some other times you can punch holes at it. Some other times you can just put something toxic in their environment and hope that they will die. So you really have to see what's the best strategy of killing the enemy, okay? As I mentioned, one of the strategies is to eat them up. So that is phagocytosis. So you mainly have two cells that are the big eaters of the immune system, okay? One of them is your neutrophil. And then the neutrophil is really patrolling things in your blood. So neutrophil is floating around in your circulation, okay? And the other major eater is your macrophage. So the macrophage really likes to patrol tissues or organs. So you have some macrophages that are called fixed macrophages, and those like to hang around things that are, just sitting there, maybe in your skin or in your liver or some organs like kidneys. They just like to sit somewhere and wait for invaders to get there. Okay, so you have the sitting macrophages, and then you have the patrolling going around the neutrophil. Those are the main phagocytic cells. All right. So here is one exercise that is connected to the Immune Quest, right? So if you're doing the Immune Quest video game, you can follow on those descriptions. Okay. All right. So here we start. How is it that you're gonna kill the enemy? Well, you can do some pathogen-directed killing. That means you can release some molecules in the environment that are toxic to the thing that you want to kill, and hopefully those are going to die, right? Besides killing the pathogen that is, just sitting there, you can also kill pathogens that are living inside of cells, like for example, viruses, right? So in that case, you have to kill the cells that are infected, okay? So you have to detect the cells that are infected and kill the whole cell, not just the pathogen. And yeah, this also means that your good cell that got infected is just going to become collateral damage, or you call it, there's another name for that, which I can't remember now. Casualty of immune war. Okay, so those are the cells you have to sacrifice to save the whole organism. Okay. All right, so concepts here. Your innate immune cell, all the cells, they have those pre-existing receptors, right? Those PRRs, and those are going to respond the same way every time it sees pathogens. All right. Or also your damaged cells, right? The detection of damaged cells is by damage-associated molecular patterns, right? So DAMPs. Okay. And then you have your adaptive immune cells. So those are two cells that you have to think, B cells and T cells. Now, those cells, they are very specific for a type of antigen, okay? The cells that you have to think, those B and T cells, they start up as naive, so meaning they never actually saw the pathogen that they were intended to attack, okay? But then the whole development of these cells is totally different than the development of those innate immune cells, okay? The T cells and the B cells, the process they go through actually changes the sequence of their DNA, okay? So by changing that sequence, that ensures that every one of these cells is unique when it's ready to be released from the primary lymphoid organs, mainly, yeah, from either your bone marrow if it is B cell or thymus if it is a T cell. Okay, yeah.
Good question. You mentioned there's immune cells that are naturally floating through the blood or in tissues, but then there's also, it mentions, and I think this PowerPoint or the next one, that for an immune system to be effective, it has to be conducted like in the lymph nodes or in the secondary. Like, what is the difference in the responses anywhere in the body versus in those specific spots?
Oh, that has to do with the level and stage of responses. Because as we mentioned before, the first stages of response is, first is a physical barrier, right? So we have your mucus, you have the skin, you have enzymes that are present all the time. And then you have your innate immune cells. If the innate cells take care of the invader, then you're never even going to get a secondary response. But the innate cells, especially your dendritic cells, they are going to carry either the whole invader or a piece of it. They're going to carry it to a secondary lymphoid organ. In the secondary lymphoid organ is going to teach one of your adaptive immune cells, is going to teach it that this certain invader came to the body. And that's why you're going to get, the second stages.
Immune cells are only in those specific places, they're not around the body?
At first, those B and T cells, specific, they're not circulating. They never saw antigen, so they're really naive. They're sitting in a secondary lymphoid organ, and they are just waiting to be activated. And we're also going to look at strategies of placing those organs in certain parts of the body, because they have to be really at places that are major hubs for the circulation and the lymph circulation, for everything to pass through them. Okay, so at one point, your innate immune cell is going to bring the pathogen to get in contact with one of those naive adaptive cells. Okay, so the whole response has to go in stages. Always the first stage is involving the innate cells, okay? And then the innate cell is going to teach your adaptive cells to be on alert just in case the same pathogen is seen again next time, like after 10 days or so, okay? But this whole thing is going to happen in this type of sequence, regardless of the pathogen that is encountered, okay? Okay, so a lot of times, as I mentioned, we don't really know what is really the effectiveness of innate cells because we never really get to see the pathogen. It doesn't survive the first lines of defense, so we have nothing to work with. So that's why it's a bit tricky to really understand what's happening, and especially when you get to gut mucosa, and that's like, oh my goodness, because a lot of times we can have similar cells that we can use animal models, like mouse or rodents or some other type of animal models, but in case of gut immunity, the cells are different. You can't really base the response from a rodent to our own response because they're not even the same cells. You can't work with them. It's kind of very, very tricky. Okay. All right, so main idea here, those cells are always responding the same way. They're the first ones into battle. And then later you're going to have activation of those B and T cells. And those are very specific. And the way that you get so many specific T and B cells is really because they rearrange their DNA. Okay, so each B and each T cell has a very unique genome. It's not the same genome that you were born with. Okay, it's very unique to you, and it's not the same as the somatic cells or all the other cells in your body. Okay. So here, this whole idea of receptor selectivity and how is it that adaptive immune cells get to respond, better to a second time that this is a pathogen, right? We have to keep in mind that the first time this is a pathogen is really called priming, okay? So the cells have to be primed. And then the second time this is a pathogen, that's when the cell is already, ready to respond, okay? So some scientists, which I never ever asked about names of scientists because I never remember them anyway, okay? So those first scientists, they came up with this idea that not only pathogens, but pieces of the pathogens that they called non-infectious materials, they could also teach those adaptive cells how to respond, okay? So they came up with this initial theory that they called side chain theory, that they said the following here. The first time that this adaptive cell, so this is, let's call it a B cell, okay? The first time that the receptor gets activated by some type of stimulus, right, either pathogen or a piece of the pathogen or non-infectious particles, right, which could also be called some type of vaccine, right? So the first time that it gets activated, what happens is that receptor, okay, first time is getting activated, that receptor causes the cell to produce more of its kind. So after attachment of this first thing, antigen, then this type of receptor starts to pop up in the cell surface, okay? So these guys were thinking that if the cell has a lot of these receptors popping out, so when it finds more of the antigen, it's capable of binding to the antigen in a much better way than if it had only one receptor. Okay, so this was that initial idea of selection of receptors that can react to a particular antigen. So they were trying to explain that second response of the adaptive immune system that is bigger and better than the first response, right? They were trying to explain memory of the adaptive immune system. And they were really in the right path. They, imagine that this is like early 19th century, something like that. It's 1900s, maybe late 1800s, something like that. So they didn't have a lot of tools to study this, and they really had to use their imagination. So this is really the best they could come up with. All right. Later on, this initial side chain theory was more refined into this clonal selection theory. Okay, so what does that mean? Okay, when you compare to the side chain theory is that several cells have receptors. If the receptor gets selected because some antigens showed up, then the cell is going to make more of that receptor. Okay, initial idea is fine. Now, when you look at actually what happens is that each cell has a lot of copies of the same receptor. Okay, so this has to do both with the B cells and the T cells. Each B cell has a lot of only one type of receptor. It's very, very specific. Okay, now if you have an antigen that is specific for that receptor, if you have the antigen that binds the receptor, is not only the receptor that it starts to pop out of the membrane. That doesn't happen. That cell already has many of those receptors. But what happens is that the cell gets triggered to start dividing. Okay? So that one cell that was very specific for that one trigger is going to start to proliferate. Tongue twister. Okay, it's going to start to divide very quickly. That division is what you call clonal selection. So it's called expansion or clonal selection. Why? Because there was one clone of that one receptor that triggered division of the cell, and now we're going to have a lot of cells that are going to be able to attack that one type of antigen, which is a good idea because those antigens, they're coming with the pathogen, and they usually, they don't come alone. So if you have an infection, you're not going to have this one lonely bacterium floating around. You're going to have a lot of those bacteria floating around, right? So you want to mount a lot of cell response to go there and kill all of this invading army, okay? All right, so this is the idea of clonal selection and clonal expansion. All right, so here the picture from your book showing that you do have B and T cells, that they are coming from the stem cell. So that's your pluripotent stem cell that we're going to look more at today. Okay, coming from your stem cell, and you're going to go through a process in the primary lymphoid organs that is going to rearrange the DNA of these cells and is going to put some type of receptor on their surface. Okay, so if the receptor is useful, then the cell is able to pass through first stages of selection. If the receptor is not useful, then the cell is going to die, okay, in the primary lymphoid organs. It will never come out into the circulation, right? So if passes the selection process, then it goes into the circulation. Now, let's see this cell here. In the circulation, it found its antigen. This is when the cell gets triggered to start dividing. So it's the same type of cell that divided, and now you have that clonal expansion. So that is going to happen in the secondary lymphoid tissue. Okay, this detection and expansion is going to happen in secondary lymphoid tissue. So make a note here, because here I had primary one. Okay, so this is a primary, and this is your secondary lymphoid tissue. Okay. All right, so what allows the B or the T cell to recognize pathogen or antigens with so much specificity? Okay, so you have those receptors that are characteristic of each one of those. You have your B cell receptor, and then you have your T cell receptor. Okay, the way that those receptors come up is because of gene rearrangement, meaning shuffling up the DNA. Okay, so you should you should be used to these things by now, that if I have something in red and bold and underlined, it's probably very important, and that translates into test questions, right? Okay, so B cells have those B cell receptors, and those B cell receptors are also called antibodies, okay, and they are also called immunoglobulins. Okay, The T cell receptor, you just call it a T cell receptor. Okay? Major differences is that B cell receptors, they have a lot of different types of these receptors. They can be attached to the cell, and then after the cell gets activated, the B cell can start shooting out receptors so they can detach from the cell surface and then go and see and attack the pathogen. Okay? One important thing about B cell receptors is that they recognize the antigens floating around by themselves. Okay? So B cell receptor is capable of going around feeling the environment and recognizing the receptor in the native state, meaning the whole protein or the whole thing. It recognizes the whole thing. Now the T cell receptors, they are not capable of doing that. And we're talking about T cells that are systemic, meaning goes around in your circulation. Okay? Those T cells, the T cell receptors, they need another protein that is present in a presenting cell. So the presenting cell of your innate immune system is going to come with a certain type of protein and show a piece of the pathogen to those T cells. So T cells really depend on presenting cells, and the presenting cells have these special proteins that you call major histocompatibility complex, or MHC proteins. Okay, so this is one of the major differences between T cells and B cells receptors. B cells recognize the 3D version of whatever it is, okay, or the whole thing. And the T cells, they really need something that is presented to them in a platter. Okay, major difference. Okay. So here comes a fun addition to your slides, right? Again, how is it that you're going to attack your invaders, right? How is it that you're going to try to kill the thing that's not supposed to be there, right? So you have to elaborate and expand your defense, right? So this is under that aim of functional organization of the whole system. Right? So there are two ways that you can attack your enemy. First way is cell-to-cell contact, right? So you go in this mano a mano type of fight. We have a cell here, you have a cell there, and they fight. Easy to understand, okay? So you have, in order for this fight to happen, you have to have some recognition, you have to have signaling that is going on, you have to have the cell activation, right? So it is a more direct touching each cell type of contact, of fight and killing. And on the other hand, you can have secretion of molecules in the environment, and you hope that whatever you're putting out in the environment is going to kill the thing that is going to touch. So here is the analogy of this other form of attack. You drop bombs and hope the bombs are going to kill only the enemy, right? But then again, with the same analogy, the bombs are dropping also in territory that you can have your friends around, so it could kill a lot of things, not only the enemy, right? Usually those bombs are kind of deadly, okay? So these things here that are things that are dropped in the environment are very much related to what we call humoral response. Okay, so those are antibodies or some proteins that are floating in the blood. Okay, so humoral response. When you have this type of cell-to-cell contact, that is called a cellular type of response mainly. Okay. So here, when you look at humoral responses, okay, so here they have the examples of B cells and T cells. Okay, so this is talking about adaptive immune system type of response. Okay, so when you look at humoral response of your adaptive immune system, you are going to have antibodies. Okay, so humoral response means the bombs dropped off from this thing, and those bombs are your antibodies. All right, proteins floating around in the circulation. Okay, when you talk about cell-mediated response of your adaptive immune system, that really has to do with your T cells. Okay, so T cells are going to do contact-based type of signaling and killing the target. All right. And here is your first question. So T cell receptors and B cell receptors are what? Remember, this is the time that you can consult your neighbors.
It says it right there.
Yeah.
It's got.
So time is up. What is your answer? Is E. I see several people answer C. C says receptors found on all immune cells. No, those are B or T cells. Receptors, they're not in all immune cells, right? When we talk about all immune cells, those are including also your innate immune cells, right? So those are not in innate immune cells. Some people answer B. T cell and B cell receptor are not pattern recognition receptors. Those are for your innate immune cells, okay? So that's pattern recognition receptor is, we don't talk about pattern recognition when you talk about B and T cells. You talk, B receptor or T receptor. Because remember, the pattern recognition receptors are coming from your innate genome. It's not going to be rearranged. You only have a few of those that are expressed, like hundreds of those or so, comparison to millions of possible B or T receptors. Okay. All right, so now you saw the pathogen, you attacked the pathogen, you mounted this huge immune response, and then you killed the pathogen, you eliminated your enemy, and now what? Right. So now you have to stop the response, right? You have to quiet down your immune system, because if you don't, then you're gonna have problems, right? Overactive immune system causes a lot of problems, okay? So what do you do? How is it that this is happening? So if you have a pathogen present, that means you're stimulating your immune system, okay? The stimulation is the presence of the pathogen. If you don't have the pathogen, your immune system doesn't get triggered, okay? And that makes sense even if you use an analogy of other proportions. Like, for example, if you have, let's say, one person that pushes your buttons, and there is always that someone, right? Like, I tell you, I tend to be a very peaceful and accommodating and understanding person, but I had this one neighbor, I tell you, if I tell you, you wouldn't believe it. Okay, but anyway, he pushed my buttons, like, really, okay? Anyway, ended up that the guy was forced to move out of the community. Yeah, he was that bad. I can't even tell you, start telling you what the guy was doing. So he was forced out of the community, but every time that I would see his car, he was like stalking me, okay? Every time I would see his car, my blood pressure would go up. Not only me, but I had my dog, my little dog. He was a puppy when this guy started with this whole nonsense, and the dog started going crazy every time the car would come around. But the dog would feel that I would get nervous. He look around and say, oh, the blue car, it must be him. So now I have my primary response and my secondary response, And the guy would come by and like, I just wish I could just punch him, cell-to-cell contact. That's it. And then I'm thinking, maybe I'll just blow up his tires or something, you know, like humoral system, you know, just drop some nails on the floor and poof, you know. But I had all these ideas of how to attack the enemy, okay? And then he moved out. I had nobody to fight with, okay? I mean, the guy was gone. And I'm like, peace and love again, and everything is cool. And except that every time that my dog would see a blue car, he would get nervous. That's the memory of your secondary immune system, okay? But, you know, adaptive immune system, right? But then if somebody you're fighting with is not there, who are you going to fight with? That's a problem. You don't have anybody to fight with. I mean, if you start fighting with yourself, that is a dysregulation of the immune system, overactivity. You don't want that to happen, right? Same thing here with your cells. If the presence or the stimulus is not there, then you have to quiet your cells down, okay? So in order for the cells to relax, they have several mechanisms, as we're going to see more details later, but one of the mechanisms is that the cell just tell itself, you know, look, nothing is stimulating or pushing my buttons. I'm just going to relax. I'm going to chill out. Besides that, okay, that's called the tolerance, okay, and we're going to see mechanisms also later on, right? Besides that, what can happen is that you have specific immune cells that are intended to be the peacemakers, okay? So those are your T regulatory cells, right? Because you don't have chronic inflammation going on. You don't want to have chronic inflammation. You don't want to have overactive immune system, okay? So the cell can either quiet down on its own, or you can have peace keepers that go around and tell everybody to quiet down, okay? So those cells, the natural peace, you know, they have a little you know, flower crown peace type of thing. Those natural regulatory cells, they are type of T cell. Okay? They're called T regs. And just so you remember what they do, I put a loving T rex here, you know, peace and love, you know. Peace, love, T rex. I mean, think about lovingly T rex. I know it's a bit counterintuitive. Those teeth especially don't look very loving, but yeah, the peaceful T-regs are gonna keep everybody quiet. Like how? Well, they're going to secrete certain cytokines, certain proteins. They're gonna tell your active cells to relax, right? So those proteins are gonna put those active cells into some kind of antisocial mode. Okay, now, even from this early stage of understanding these mechanisms, there are certain proteins that you should keep in mind, like from the beginning, okay? A couple of these quiet down type of cells that you have here, I mean proteins that you have here, one of them is TGF-beta, okay? And the other one is interleukin-10, okay? Now, there are ways that you can remember what the functions of these proteins are. Okay, if you think about TGF-beta, you can think like TGIF, I'm gonna chill out, right? Thank God it's Friday, right? TGIF, okay, time to relax, all right. And if you think about interleukin-10, you can think about 10 fingers, like relax, calm down, you're gonna suppress something, right? So interleukin-10 and TGIF, I mean, just TGF-beta, all right? Okay, so what do these things do? They are going to keep those cells in the antisocial mode. So everybody cool with that? I hope. Okay. So another mechanism that quiets down cells that could get activated or were activated is this idea of tolerance. Okay, so your immune system it has to be taught on how to not destroy your own cells and not to go crazy, okay? So this idea of tolerance is going to avoid self-destruction, okay? And let me see here. Yeah, also when you talk about tolerance, you're really talking about your adaptive immune system. Those are your B and T cells. So they have to learn what is self and also what is non-self. So they will have to learn to attack only non-self, and they have to be tolerant to the cells of your own body. And that includes your microbiome, okay? Your microbiome is part of you, although they are bacteria, fungi, viruses, but they're also part of you. They cannot be destroyed, okay? So your immune system has to learn how not to destroy those microorganisms as well. Okay. Okay. Here, what is important here? Yeah, I already said that. All right, so when the activated clonally expanded immune cells, when they have to quiet down and they don't have any more stimulus, right? So they have to undergo clonal deletion. They have to disappear. And how is it they're going to disappear? Is not the magic trick. They have to die, okay? The way that they die is by a pathway that is not going to cause inflammation. Okay? And that pathway is called apoptosis. It's programmed cell death. Okay? So how you get rid of the cells that are not useful now because the pathogen is gone? You're going to let them die peacefully by apoptosis. Okay? Okay, and then you start getting here in the, to wrap up this overview, you start getting into the idea that not everything works perfectly fine all the time. And that is the part of immune dysfunction or pathology, okay? So what happens? Sometimes when your immune system is too active or hyperactive, this is when you develop allergies. Okay, there are several types of allergies. So you have, mainly like 4 different types of allergies. And besides allergies, you can also develop autoimmune diseases. What is the difference? In allergy, your cells are going to attack something that is foreign but is harmless, like pollen in the air. Okay? When you have autoimmune disease, your cells are going to attack cells that are in your body. So it's self-attack, okay? So this is attacking something that is already there in your body and your cells should not attack. And then hypersensitivity is really attacking something that is harmless, that it shouldn't attack also, okay? Besides that, you can also have a problem or problems related to immunodeficiency, meaning that your immune system is just not working the way it's supposed to, okay? So there are 2 main types of immunodeficiencies. The first one is something that you are born with, so that's called primary immunodeficiency. That we're going to also look at some mechanisms that involve that. And in comparison, you can also have secondary immunodeficiency, and this is the type that you acquire. So mainly your immune system was working well until you acquire something that caused immunodeficiency. And in case of us humans, mainly that is caused by one virus, HIV. Okay, HIV that causes AIDS. All right, so secondary is really after some time that you acquire this from something. Okay. Now, you can see here I put the example of allergies and also of thrush. So in case of thrush, this is overgrowth of what is called a candida. It's a type of fungus that is present and it shouldn't be overgrowing, right? This is not just that little film or biofilm that forms in the tongue. Like sometimes you go brush your teeth and you're like, the mirror's like, oh, it's all white, like especially if you drink milk or something like that. You're like, oh, disgusting. So what do? You brush it off, right? Use a dental floss and you scrape it off because you don't want to go talk to somebody and they look in your mouth like, oh, all the white stuff, Okay, that's not this. This is worse. Okay, this is overgrowth of a type of fungus, right? And is common in people that are immunodeficient. Not only that, but also is common in babies because they don't have a fully developed immune system. Okay, so you find that sometimes it doesn't matter if it is a bottle-fed baby or a breast-fed baby. Thrush can be quite common with newborns. Okay, or people that really have some immunodeficient problem. And besides all that, you can also have immune imbalance, and that is the cause. That is the example of inflammation that will never go away. As we're going to see, you have two types of inflammations. You have acute inflammation that is good and should be resolved, and then you have chronic inflammation that stays on and on and on. It just gets worse. That really gets better. So this is the overall picture of immune dysfunction. Now, this is not an eye clicker. This is just a practice question that you can do in your own. Okay, but now the next one is an eye clicker, and I'm going to start it. Okay, so what is hypersensitivity? Okay, so what is your answer? Okay, everyone at once. 1, 2, 3. B. That is your overzealous immune attack on something that is not harmful. Huh? All right. Okay. And then you have these strange situations that also involve immune system. Like, for example, when you do organ transplants or tissue transplants. Now, you don't want your immune system to attack something that is not yours. Otherwise, you're going to die, right? So you have to quiet down your immune system so it doesn't reject the organ that was transplanted or the tissue that gets transplanted, okay? And believe me, in order to minimize rejection, yes.
So people that I have a friend who has a pig valve.
Mm-hmm.
How does her body not react to that different foreign organism?
Okay, that is something that we're also going to see into some detail. Now, the valve itself is made up of a tissue that is very, very similar to your own valves. Okay, so it's a type of tissue that does not trigger an immune response. Okay, now for that, usually either a pig valve or a cow valve is very, very similar to our own valves. The issue is that it is still not your own, so there is a certain risk that you can have some form of rejection. In order to avoid that, what they're doing lately is humanizing the pig or the cow, and pig is a lot easier to humanize, and you develop a certain valve that is exactly the same as the human one. Okay, so you have, I'm not sure if they can do that in vitro now instead of, sacrificing the whole pig. Although, I mean, if you are not vegetarian, most likely, RDP anyway, so they get sacrificed anyway. But, you have ethical issues, maybe they're not going to eat the bacon after transplanting the valve or something. I don't know. But yeah, it is a good question because when it comes to valve and heart function, a lot of times you can even have problems of your own immune system attacking your own valves if you had a certain type of bacterial infection, like a strep throat. And that can cause what is called stenosis or hardening of the valves. Okay, so that I believe I do have some mentioning of that in one of the lectures. Yeah, but yeah, it's a good question. Why? How is it that you can have transplanted valves and not a huge problem? But I'm sure that your friend, just like my mom, that had a transplanted mitral valve, she had to be on blood thinners, like forever, because there is a problem of turbulence and possibility of causing blood clots. Yeah. Yeah. All right. So when you have transplanted stuff, you don't want to activate your immune system. Okay. Now, in another situation, you actually want to activate the immune system is when you have development of some cancer cells. Okay, so now you want to get your immune system, especially your natural killer cells, to go and kill the abnormal cells. But a lot of times, the cancer cells can get away with not getting killed because they have certain strategies to fool your immune system, to go under the radar and not detected. Okay, we also have some mechanisms of how that happens, right? And also I'm going to add certain some mechanisms that we didn't know until very recently. There are certain things that you can do to avoid developing cancer cells or killing cancer cells in case you have some of them. You know what I'm talking about? No. Huh?
Stem cells?
No.
CRISPR.
No. Something really simple. No. Yes! Exercise! Believe me, you exercise, you are going to kill some cancer cells. Because your muscles, you're exercising muscles, not your sitting couch potato type of muscles. Those are not doing anything, like ever. Right? I even doubt if you have certain muscle mass just by sitting and watching TV. Oh, sometimes that remote muscle. That doesn't count. Okay? Video game muscle doesn't count. Right? Exercise, big muscle type of exercise. You can kill cancer cells. There are some proteins that your muscles make and put in circulation. Those are called myokines. Sounds like cytokines, but they're myokines. They kill cancer cells. And I can tell stories about myokines that we didn't even know existed, right? I'll tell you that when we get to that part. I tell you, I'm always full of stories, so I always bring myself back to the class, right? All right. So now we're going to chapter 2. We finished this whole overview of the immune system, right? And I know there's a lot of information, but the main idea that I really want you to get from the last, 2 classes today is how is it that the whole system is acting? Okay, the whole body, you have this outside protection, you have this innate form of defenses, you have the barriers. If the invader goes through, then you have the innate cells that jump into action. They can do cell-to-cell contact, they can punch the invader, they can drop stuff in the environment, right? So you have cell-to-cell, you have humoral, and then you're going to activate your adaptive cells, and then those are going to come into contact, come into action like second time you see the same invader. So you have all these things happening at the same time. Okay, so now we're going to start looking at specific functions of cells, starting with your innate immune cells. So what is it that they have, how you can distinguish them, and how you can identify which one is doing what, and what are the most important mechanisms that they are using for the job that they are doing. So this link I had seen before. There was this Japanese cartoon. There was this, cells at work. I was trying to go through the link and see if it's still working, but for some reason my internet is not working. Is it only me? I mean, do you guys have a problem with the internet today? Yeah. Like I tried to use Chrome and it's like it wouldn't work for anything. And the other one that I don't like, Explorer, and it's like, I forget it. I'll just test it later. But try to see if the link is working, and if you like to watch cartoons, they are actually very precise in their mechanisms. So I really like those. The only problem is that sometimes they are in Japanese with English subtitles. If you speak Japanese, that is advantage. Okay, so here let's start looking at the cells. All right. So all your blood cells, meaning and the cells of the immune system, right? They're going to come from these stem cells. So those are your pluripotent stem cells. So these guys can become any of these other cells that are under here, okay? So you have two main lineages that the stem cells can undertake. Now, these stem cells, they are going to be sitting in a specific organ in your body, and that's in your red bone marrow, okay? So red bone marrow. When you look at children, that red bone marrow can be in long bones, like femur and humerus, right? But when you look at post-adolescent and adults, those red bone marrows are going to be in flat bones, or non-formed or strange formed type of bones, okay? So the pluripotent stem cells are going to sit in the red bone marrow, and depending on the signal that they get from the environment, they can divide. One of them stays there, another one can go into one of these two lineages. So the myeloid lineage mainly is talking about all the cells here and other cells here, all the cells that are involved in innate immune system, including the monocyte right there. Okay, so this is myeloid, dendritic cell, macrophage, all these guys here. I'm gonna go in yellow here. All these guys here are myeloid. Okay. And the other lineage that we have is the lymphoid lineage. So that has to do with your B cells and your T cells. So those are your main cells of your adaptive immune system, but also your natural killer cells, right? So your lymphoid is B and T cell and your natural killer cells. Now the natural killers, those are part of your innate immunity, okay? But they are coming from this lymphoid lineage. So what do you do here? You memorize the ones that are coming from the lymphoid lineage, because there are only 3 of them. And if it is a type of cell that is not one of those 3, so you can bet it's coming from the myeloid lineage. Easy? Okay, so you got your lymphoid and myeloid. All right. Now, when you look at your myeloid lineage, keep in mind that also you have your red blood cells and your platelets that are also considered as formed elements in your blood, okay? Those are not really cells of the immune system, okay? But they're in the blood, all right? And everything has some relation to immune system function, right? So we're gonna see more on those later as well, okay? All right, so innate immune system cells, and then adaptive, those are your lymphocytes. So that's easy. Lymphoid lineage gives rise to lymphocytes. All right, starts the same thing. All right. Now we're going to look into more and more details on the differentiation of these cells. All right, so here's where we start. The stem cell right here. First cell that is capable of differentiating in all these other guys. So that is your stem cell. We call this also hematopoietic stem cells, because hemato is for the blood, okay? So this guy here, it says is self-renewing. What it means? It means that when it divides, one of the cells is gonna go towards a lineage, but the other one is gonna keep on being a stem cell. Because imagine that when it divides, if both cells will go towards some lineage, then you're not going to have any stem cell left, okay? And you need to have stem cells left. So it's these ones, one of them, of the daughter cells, is going to go towards a lineage, but the other one is going to keep on being a stem cell, okay? All right, so let me see here. When you look, there's a lot of information in this slide, okay? When you look at your lymphoid lineage, you'll see that it is possible that some dendritic cell can derive from this initial stage of differentiation, okay? Yes, it is possible. And the dendritic cell is not B or T or natural killer, so dendritic cell is part of your innate immune system cell, okay? But this is not what usually happens. Usually what happens with dendritic cell, usually they're coming from the myeloid type of lineage, okay? Either directly from the precursor or from another type of cell that is a monocyte that can also differentiate into the dendritic cell. The dendritic cells are really fascinating. They are crucial for activation of your adaptive cells, okay? And they're very crucial for initial response and activation of your adaptive cells. So let's look here in your lymphoid lineage, okay? This here is happening in the bone marrow, right there. Okay, so this is in the bone marrow. So something is going to happen, something in the environment, like some kind of cytokine or growth factor or transcription factor, is going to be present there, depending on what your body needs, that is going to tell this cell to go down this lineage. Now, once you have a precursor of a B or T cell, what is going to happen is that precursor is going to become under development or further precursor of B or T cell. And if it is a B cell, is going to further develop in the bone marrow, that's the blue part, okay? In the bone marrow is going to develop in your B cell, okay? Is going to learn what is self, what is non-self, is going to develop the receptors, the B cell receptors, and then once it's finished with that differentiation, is going to come out of the bone marrow and is going to go into the circulation, meaning it's going to go into the periphery. So that's your B cell. The whole development happens in the bone marrow. When you compare that with a T cell, what's going to happen here is that the T cell is going to start developing in the bone marrow, but then it's going to travel to the thymus and finish the education and the development of the receptors inside of the thymus. This is why the thymus is considered as a primary lymphoid organ, okay? Because the development of the T cell is going to happen there, okay? The development of the B cell is happening in the bone marrow. Development of the T cell is happening in the thymus, okay? Now, after it develops certain types of receptors, then this T cell can become either a helper cell or a cytotoxic cell. And of course, we're going to see a lot more how that happens and what are the functions of those cells. All right. Now, besides those two cells, you also have your natural killer cells, or also called immune lymphoid cells, okay? And those coming from the same lineage. So develop in the bone marrow and they go to the periphery and they do their jobs. Okay. Now, when you look at the lymphoid lineage, okay, so you have your, myeloid lineage. So now you have the precursors for all these other cell types. All of them are going to develop in the bone marrow. That's it. When they come out of the bone marrow, they're ready to attack. Yeah.
So you mentioned the cells divide as well. So each time the cell needs more of each type, it doesn't need to go through the whole process, the stages. It can just divide when it has one already made. Is that correct?
That is mainly for your B and T cells. Okay, your innate immune cells, they are going to come from the bone marrow, and once they go into circulation, they are not going to divide.
After some certain amount of time, even the B and T cells must die automatically and need to be remain. Is that
Yeah. Yeah, what happens here is that if you have a clonal selection of B or T cell, then if you don't have any more the pathogen, then you don't have any more use for those cells. So when you talk about clonal selection or clonal deletion, you're really talking about B and T cells. Yeah, because the other cells, they're going to divide and differentiate in the bone marrow. By the time they come out, they have a very short lifespan. And they don't divide out in the circulation. They just do their jobs and die. Or they just die without doing their jobs just because they get old. Kind of depressing. But yeah, but that's the reality. Okay? All right. So properties of your hematopoietic stem cells. I have what, 9 minutes, maybe a little less? Okay, so good. Okay. Properties, they have to renew themselves, right? They have to give rise to all the other cell types. And let me see here. Okay, I already mentioned that. Usually they're quiet if your body is just not fighting any type of invaders. All right. So now we're going to start getting into more details on how this differentiation happens, right? So depending on certain proteins, they're called transcription factors. Depending on the presence of those proteins in the bone marrow, red bone marrow, okay, that's going to tell your hematopoietic stem cell to do one thing or another. Okay, so even from these early stages, even before we get to more and more details on differentiation and regulation and all that stuff, you should start keeping certain things in mind. Like, for example, if you are talking about Icaros or GATA3, you are talking about lymphoid lineage. Okay? If you're talking about GATA1, you are talking about myeloid lineage. Now, you find whatever mnemonics helps you, but I always have some little issue here on trying to remember GATA1. GATA in Portuguese is a cat, and that didn't make sense. And I'm like, okay, just remember, just memorize it. Okay, fine. So let's say you have transcription factors here that are important for your lymphoid lineage. So now you're going to commit certain cells to go down that path. And then look, further down, you also have GATA3 over there, okay? That is going to further differentiate your progenitor into a type of T cell, okay? So this is like, you're starting to think that depending on the transcription factors in the environment, you're going to have cells that are going to be pushed one way or another. All right. So what are those cells and what's in the blood? Okay. So those cells are circulating. And when you look at your blood, and let's say here you spin down the blood, you're going to see that most of your blood is made up of plasma. That's about 55% of the volume, okay, if you are well hydrated. Okay. So most of the blood is plasma, and here in the plasma, you're going to find proteins, you're going to find antibodies. So your humoral defense is going to be here in the plasma, okay? Proteins that are made by your liver, and that's your, what's the name of that? Complement systems, okay? And then antibodies made by your plasma cells or B cells that were differentiated, going to be there. Now, when you look at the portion that is your red blood cell, that is about 45%, and these are all red blood cells, tiny, important red blood cells. In the middle, you have this little percentage here that you call the buffy coat, and this is where you're going to find all of your white blood cells. So that's going to be all of the cells that came from lymphoid as well as myeloid lineage, and they're all looking white. So that's why when you look at the cells in the buffy coat, they're all white. Call them leukocytes. That means white blood cells in Greek. Okay? Okay, now, when you look at the composition of your white blood cells, there are certain telltale signs that you can see, and you can start predicting what's going on with the blood, with your system, right, with your body. So when you go and you don't feel well, you kind of have a fever, you go to the ER, they first thing they do, they poke you. Hopefully they know how to get your vein, and they'll start poking and digging and poking and digging. And oh my God. But anyway, so they say they get your vein, right? They spin it down, they look at the white blood cells, and they see a lot of white blood cells. That is not good. That is telling them you get some kind of infection. What kind of infection? Usually is some kind of bacterial infection. Okay? Now, let's say that instead of seeing a lot of white blood cells, let's say that they found not enough or not a lot of white blood cells. That is usually a telltale sign that you have some kind of viral infection. Okay? Just, you know, there's just a ballpark type of ideas on this. Okay. So, all right, so I'm going to stop here and pick up on different types of innate cells next time. Remember, I'm going to put the video, okay, for Thursday, because I will not be able to be here on Thursday. Yes, this Thursday.